Okay, so almost 9:40. Let's have a class. Today's fact sheets will be here, and it's your last Tuesdays, and we will try to finish the biochemistry test. So we're going to talk about today is started from the chapter 16 right here. Okay, we're going to talk about most of these tests except the nitrate reduction. We're not going to do that one. But most of the others we will do. Today, basically, we do the inoculation, and next week, Tuesday, we will do the confirmation. Okay? So that's being said, it's, I think it's lab 12. Biochemical test 2. Okay, remember, we already did a 2 biochemistry test. 3 tube fermentation and urea test. That's before the throat culture. So today we're going to continue to do some. Okay, let's say number 3. That's called the SIM test. Okay, SIM test. It will be using needle instead of loop. This is important. So where is the needle? This is a needle. This is the only time you're going to use in the lab, use a needle, the sharpie end. This is what you traditionally used is a loop. Okay, so today for this specific test, we do not use loop. We're using needle. Okay, how to do it? You will get a medium. And then using needle to pick bacteria. And then right in the middle, you go down and then go up. So first of all, this is a semi-solid medium, which is containing roughly 0.15% agar. It's very soft, and I'll tell you the reason later on. And using this needle right in the middle, back and forth one time, we are talking about is 1 stab, 3 jobs. So what are the 3 jobs? Corresponding to SIM, the SIM test each represents an index. S means sulfide. Some of the bacteria can use sulfide and reduce to H2S, hydrogen sulfide. This is we are talking about is anaerobic respiration. We will talk in the lecture soon. Okay, what is called aerobic respiration? What is called anaerobic respiration? What is called fermentation? This is a part of the anaerobic respiration, which means the bacteria using electron acceptor is not oxygen, is other inorganic chemical. Once it's reduced to H2S, it will turn black. The medium will turn black. Now, of course, this has to be incubated 35 degrees Celsius at least 24 hours. Now, why it will turn black? The reason is there is a metallic ingredients such as iron in the medium. And once you have H2S generate, it will turn black. So, on next week, Tuesday, if you see your SIM agar turns black, which means it is sulfide test positive. Otherwise, it is negative. So that's the first S. Okay, what is I? That means Indo. So what is Indo? This is byproduct of tryptophan. Is an amino acid, essential amino acid. So what it is looks like. Goes here. Okay? This whole thing is tryptophan. Remember, chiral C in the middle, amino group, carboxy group. Universal have an H there, and this is all side chain, and this is called tryptophan. So why we say the byproduct of tryptophan? If bacteria can use tryptophan, it will be, not here. It will be split from the middle. And this part is Indo. Now, how do we know bacteria created Indo? After incubation, 35 degree, 24 hour, we need to add a reagent on the top. The reagent is called Kovac, like this. At the very top of the SIM agar, very quick it will turn cherry red. That's why it's red. If it turns cherry red, it is Indo positive. So we add Kovac reagent. If it turns cherry red, it is Indo positive. Otherwise, it is Indo negative. And I want to say very straightforward to you. Indo test is a very easy, straightforward test to testing a bacteria. Everybody knows it is E. coli. A very quick test. So the rapid testing research area has been developing different methods specifically for E. coli. One of the methods is testing Indo. If the bacteria generated Indo, it's very possible it is E. coli. Okay, so that's I. Now, what is M means? You should know what is M means. Motility. How do we know bacteria has a strong motility? Bacteria need to have flagella. So motility test is relatively straightforward. After your inoculation, you will see after incubation, 35 degrees Celsius, 24 hours. If the bacteria just stay in the middle, which means there is no motility. In other words, if you see the bacteria goes everywhere, That is motility positive. In other words, it's very possible it has the flagella arrangement is petrotrichous. Remember flagella we talk about monotrichous, amphitrichous, lophotrichous, and petrotrichous, which means a flagella goes everywhere on the surface. There is a pathogen is very typical for motility positive is Proteus vulgaris. We used to do this in the lab. We are not allowed to do because it's soil pathogen. And today we'll use citric acid bacteria, citrobacter, as a replacement, as a surrogate. Okay, so said very simple. SIM test. You came here to get a SIM solution tube. And then use a needle to pick your bacteria. And then very gentle, strict stab down, stab out. Right in the middle. Then we will just put on your tray to do the incubation for 5 days. So instead of 35, 24 hours, we're going to all do today is 22 degrees Celsius, 5 days. And on Tuesday, we need first to look at motility. Now, by the way, this is the reason why we use semi-solid media to test the motility. If you see the bacteria right in the middle, no motility. If you see the bacteria goes everywhere, motility positive. Second, we're going to look at the color. If you see this turns black or even partially black, we say sulfide positive. Then we're going to add is Kovac reagent on the top. I'll just do a demonstration for a couple of samples, not everybody, okay? If you see cherry red color comes out, it is positive. And I can tell you right now, E. coli is positive because I want everybody to know it's a very quick test for E. coli and very useful. And I also gave you an example. This work is simple. You can do it in 5 seconds. But the story is this long. So you need to know, okay? Same as a lecture, as a lab, we're going to talk lots of chemistry. You're going to see me draw the whole blackboard today in the lecture, in the next 5 lectures. So we're going to talk lots of the chemistry. And I also want to fix some of things you haven't been learning a lot in your biology class or even in the other classes. I want to make sure you understand. So that's why we're going to talk lots of chemistry. Okay, so that's first one, SIM test. Second one, the aminase test. The aminase test is also very simple. You're going to get a slant called the aminase slant. And then you inoculate the bacteria, but this time use loop, not a needle. So, what's that? You'll get a slant. The slant called phenylalanine deaminase slant. And then you inoculate the bacteria there. And then we're going to incubate 22 degrees Celsius for 5 days. And then what we do, we add a special dye called ferric chloride. If you see it turns dark green color, which means the aminase positive. Okay, now the story is, what's the principle behind it? You need to know what is phenylalanine. So what is phenylalanine? Let's talk about it. Same thing. This is major structure of a general amino acids. Amino group, carboxyl group H and R. For alanine, this R is CH3. And you know that. What we call here is phenylalanine. What this means? We add a phenyl group there. So this becomes 2. Then on the top, you have a phenyl group there. That's phenylalanine. Now what means deaminase? Where is the amino group? It's right here. So what this means? Which means in certain circumstances, bacteria can use amino acid to survive. And once it's using amino acid, this deaminase, this is gone. So the rest is this guy. What is this? Do you know this? What is this? This pyruvic acid. Then what is that? Very similar. Phenylpyruvate. What is pyruvic acid? The one after glycolysis. Is that correct? What this means? What means phenylpyruvate? It's a modified pyruvic acid, which means in some environments, bacteria not use glucose to go through glycolysis. They use amino acids, which is the phenylalanine, and generate the phenylpyruvate. And this is, we call it intermediate metabolic products, and or precursor, we call it. It will can directly jump in the next biochemical reaction. What's that? Citric acid cycle. But of course, it's a modified citric acid cycle. Is that right? Because pyruvic acid goes to citric acid cycle. And this phenylpyruvate, very similar, and the phenyl group goes to modified citric acid cycle. And bacteria can survive. So that's why they do deaminase. Okay? So that's called deaminase test. That is number 4. Okay, number 5. Oxidase test. So what is oxidase? Oxidase, we have to talk about the electron transport chain. We're going to talk more detail in the lecture. So here we're going to be briefly. So you have NAD, FeADH2, NADH2, then you go FMN, FES, then you go coenzyme Q, you go cytochroma C, cytochroma A, cytochroma A3, and finally goes to oxygen. Remember, this is in your biochemistry. That is a electron transport chain for most of the bacteria is using. And we'll talk about this in the lecture. Okay, all kind of story there. For some of the bacteria, once you goes here, you want to continue to transfer electron. You need an enzyme, the enzyme called oxidase. So, oxidase originally is called cytochroma C oxidase. But again, people think you are very smart, so they took this out. Become oxidase. Now, how to do oxidase test? That's very straightforward. You get a neutron agar. On the left side, you inoculate the bacteria. On the right side, you inoculate another bacteria. And this time, we're going to inoculate E. coli. And here inoculate Citrobacter. Then we incubate 22 degrees Celsius 5 days. And then we add oxidase reagent. Oxidase reagent, the name is very long. I'm not going to talk about it. I don't even know. It's very, very long. Like 20 letters the name. If you see it is blue to purple color, it is positive. Otherwise, it is negative. But be careful. Oxidase test you have to do within 1 minute. You cannot do more than that. Why? Our environment has oxygen. If you open the agar plates exposed to the air about like 5 minutes, everything turns false positive. Because we have air. We have oxygen in the air. Okay? Now, why oxidase test is important? This is used to differentiate an important soil bacteria, also sporage bacteria, to the other group of the bacteria. This is differentiate Pseudomonas versus a group of bacteria called Enterobacteria ACA. Pseudomonas, for example, Pseudomonas aeruginosa, this is also a gram-negative logs bacteria. And the Enterobacteria ACA basically is gram-negative logs, gas fermentation positive, acid fermentation positive. It is also, we call it a facultative, and the non-spore forming. Non-spore forming. What this means? Which means if we do gram stain, looks very similar sometimes. That's why we do an oxidase test. Plus, Pseudomonas is a strict aerobic bacteria. They must use oxygen. They must going through the standard electron transport chain. So, Pseudomonas is oxidase test positive. This whole group is negative. What are the other bacteria or oxidase test positive? How did you do for the exam 2 lecture? I saw somebody take in 20 minutes. You should take in 10 minutes. And you stuck there for 1 hour, 1.5 hours, you're not going to do very well. I'm telling you. So, which bacteria oxidase test positive? Neisseria, is that right? Neisseria gonorrhea or Neisseria meningitis. Especially gonorrhea, we said we know the people do the sexual activity. Why? Oxidase test positive, turns purple color. So that's something we're connected to the lecture, just want to let you know, okay? Oxidase test. Last one. MRVP test. That's the last one. Okay, no more. Because otherwise you're going to be confusing. And no more blackboard. So that's the last one. MRVP test is a group of the test called IMVC. That is part of that test. In the food microbiology area, this is a very important experimental quick testing. We use IMVC test to differentiate two important bacteria, which is E. coli and Enterobacter. Because the results is very interesting. For E. coli, is positive, positive, negative, negative. For Enterobacter, is negative, negative, positive, positive. What this looks like. sticky ends, remember? DNA sequencing, okay? So let's talk about that. What are these things? What is indol? What does I means? I is indol. We already talked. So I is indol. What is N? This is very confusing one. This M here refer to MR, methyl red. This M here is not motility. That is methyl red. What is methyl red? What means the test? Methyl red test is used to testing fermentation process created acid products. So the experiment is very simple. Today you get a MVP reagent and you add a bacteria there, E. coli or Enterobacter. And then we do the incubation, and after that we're going to add a reagent called a methyl red reagent. Let me find it for you. Yeah, methyl red reagent. We add a couple of drops. That will be next week. If you see this tube turns red color, that is methyl red test positive. What this means? Which means that during the bacterial fermentation process, they generate acid products. So that's called a methyl red. Okay, what is V? That is VP test. So V here represents VP. What does VP means? That is not vice president. Vocus Prescia. What is Vocus Prescia test? Fermentation could generate acid, but fermentation sometimes will also generate a neutral products. So they first generate acetone and then become diacetone. And this is a neutral products, which means they're not acid products. Remember, bacteria doesn't always generate acid because acid is harmful for themselves. Sometimes neutral products is good. Now how are we going to do it? The same tube, we pour half of them out and we're going to add reagent A and reagent B, reacted for 5 minutes. If you see it turns red color, it is positive. Otherwise, it's negative. Okay, what is C? Citrate. We don't gonna do here. Citrate test is very similar to sulfide test, where it turns black color. Black color. Okay, we skip here. So M or VP is methyl red and Vocus Prescia test. That's part of the test of IMVC. Some textbook also write IMVIC test. But never people say what does the I means. So just want to let you know those are the tests what we're going to do. Okay? So today we're going to do SIM, deaminase, phenylalanine, deaminase, oxidase, and MRVP test. How we do? Everybody come here to get a plate. Wait a second, no. Phenylalanine, SIM test. What I have so far? Okay, we have these. So came to the your desk. Okay, listen. SIM test. Remember, use needle. Okay. What you gonna inoculate? If you inoculate E. coli, You bench partner inoculate Citrobacter. Okay? That's for SIM test. Deaminase test. Streak onto the agars. If you inoculate E. coli, you bench partner inoculate Citrobacter. Oxidase test. You need to split down at the back. You do E. coli. You bench partner do Citrobacter. Don't forget the label. This one is special because MRVP is specifically for E. coli and Enterobacter. So the last test, you're gonna have to use it. These two, which means you do E. coli, you bench partner do Enterobacter. Here is not Citrobacter. Okay? Do you need me to show you? You all clear? What's the first thing you need to do? Get the slants, get the plates, have the rack. What's the next thing you need to do? Is the label. Okay, very important is the label. Yeah, go ahead and do it.